Hello, I am Grazyna Palczewska, and I established two-photon excitation to study retinal structure and function at the UCI Center for Translational Vision Research, Department of Ophthalmology. In the retina, two-photon excitation can induce emission of fluorescent light by endogenous fluorophores, including vitamin A metabolite that sustain vision, and by exogenous dyes or fluorescent proteins expressed selectively in individual retinal cells. Two-photon absorption is a very rare quantum event, so to maximize the measured signals while using minimum stimuli or imaging light power, laser light need to be chiseled both spatially and temporally. So we needed to optimize the instrument, and in such an instrument, pulsing infrared light perception stimulated by two-photon excitation of visual pigments can serve to evaluate the function of the retina, even in the eyes with lens opacities. Furthermore, two-photon excitation imaging can be harnessed, and it is harnessed in our laboratory, for intravitreal subcellular imaging of the retinal layers, including pigmented layers, such as retinal pigment epithelium. So two-photon excitation can probe components of visual systems, such as vitamin A metabolites, that in classical one-photon excitation would require UV light, which is harmful and easily scattered by the eye structure. And most recently, with the advent of experimental gene editing therapies against retinal diseases, there is a need to evaluate transfection efficiency and any deleterious impact on the retinal cell layers. Such non-invasive two-photon imaging of the eye offers high signal-to-noise ratio and the volumetric 3D images spanning the whole thickness of the retina, and serves to evaluate structure and to quantify the efficiency of the transfection in individual layers, which is critical to translate this research to studies in humans. The future is already happening. And some of the study that we have just done are the inspiration for the field. And for instance, this infrared vision is been already studying, even though we started it, it's been already studied in Spain, in Germany, and in Switzerland. And people are driving towards implementing this in clinics because it offers so many advantages. Especially you can do the test in people with retinal opacities. On the other, and also we have seen the just lately study that is also inspired by our work, where people develop not exactly two-photon excitation lenses, but the contact lenses that are based on the upconversion. So people could see in the infrared as well, even without having the power attached to the lenses. And thinking about the two-photon imaging of the retina, this is the biggest thing, is right now we are able to distinguish metabolites, like the individual particles in the RPE. We can pinpoint to them and say those are A2E molecules, those are the retinal esters, and those are maybe an NADPH. So this is already happening, and the biggest what is the future for it is really to be able to advance this to human studies.